on their way in. Okay, so good afternoon, everyone, and welcome to our Memorial Sloan Kettering Cancer Center Flow Cytometry Core Facility virtual classroom. You can see my name is Kathy Daniels, so hello. I'm the manager of the facility here at Memorial Sloan Kettering, and below me you will also see Dr. John Quinn from FlowJo. So this is going to be a joint session today, and for those of us who are joining for the first time, this is our 7th session on FlowJo analysis. And specifically today, what we're going to be going over is some high parameter data analysis utilizing FlowJo. Okay. And there have been, like I said, there are 7 sessions. So as I go along, you'll see some links where I can direct you to our previous session, going over introduction to FlowJo, specific topics, including one previous session with John where he went over an introduction to high parameter data analysis. So this is part 2. So what we're going to be going over today is high dimensional flow cytometry data, okay? And specifically what we'll be reviewing is some dimensionality reduction tools, some different clustering algorithms, and a basic review of those different methods and when it might be appropriate to use them. And of course, we always learn more than just what's in the summary, so we'll be learning more than that. Okay? Some important links as we go along. We want to thank FlowJo for their efforts, not only in helping us out with a couple of these classrooms, but also in providing FlowJo for free for researchers who are working from home during this current climate. So we thank them for providing that for free to researchers who might not have had an individual license and were working on shared accounts. Okay. Then we have the Flow Cytometry Core Facility website, which is fccf.mskcc.org. There are all the previous recordings of our virtual classrooms there, in addition to our educational content, such as our flow post-its that we carry out or post every 2 weeks, our SOPs, big variety of, or a large variety of information is there, so we encourage you to visit. Then we also have our Twitter site, and our handle is @FlowMSKCC. There we post, you know, our flow post-its there. We post any events that are coming up that might be relevant, some interesting papers to read, and more recently we've had some entertainment content where we have flow cytometry songs and poems and all that good stuff, since we love flow. Then we also have the ISAC website noted here, International Society for the Advancement of Cytometry, and specifically Cyto University, which ISAC has made free during this work from home time. So we encourage everyone to go visit that website. In addition to Flow Repository. So Flow Repository is a nice resource for anyone who might not have FCS files of high parameter data at their disposal and want to play around with some of these dimensionality reduction tools and clustering algorithms. There are definitely a couple of data sets on Flow Repository that would be beneficial for you to use there, including a recent 40-color data set, I believe, that should be going on there. Okay. Some more important links to keep in mind, Cytometry Part A journal up top, where you can find a lot of cytometry-related articles. Self-explanatory since it's in the title. And then we have MetroFlow, which is our regional New York, New Jersey flow cytometry users group. Actually, next week, we will be having a corporate member seminar series, so we encourage everyone to go and check that out. It's free for all to attend, and it's going to be 28 speakers over 5 days, including John. So please feel free to check that out. And then we have FlowTex, which is the Texas organization for flow cytometrists, and they've been putting out quite a bit of flow cytometry content during this time as well. Lastly, the last link there is just a thank you and a reference to the original dot plot where we had t-SNE looking like an FCS file, and that was from David Gravano, and you can turn text or images into FCS files, and I've definitely had a lot of fun playing around with a lot of different things with that. So if you want to take a break from data analysis and going into that, please feel free to check it out. Okay. And then really quickly, just some suggested reading that we think would be nice for everyone as a resource from today's session. There's the original paper detailing how to visualize data utilizing t-SNE. There's an offshoot of that, which was recently published in late 2019 from Annabelle Kina up in Boston, and some others describing opt-SNE, which is another iteration of that. We have the cytometry part A paper detailing FlowSOM, and a nice paper by Florian Marr, you know, asking that question, is this the end of gating, right? And going into an introduction on different methods for high dimensional analysis and automated analysis as well. So we encourage everyone to check that out. Okay. So last, I just want to say thank you so much to our group who has been phenomenal during this whole time. They've been really, really wonderful, creating a lot of educational content and doing a lot of work before, during, and I'm sure after. We have a wonderful team here, so I need to say thank you to them. And lastly, some contacts for you to reach out. So if you have any specific technical support that you're looking for answers for with FlowJo, we encourage you to reach out to flowjosupport@bd.com. If you have any questions that you want to reach out to Rui Gardner, who's the head of the facility, or myself, these are our emails right here as well. So gardner@mskcc.org and Daniel K1 at the same handle. So please feel free to reach out to any of these if you have any questions, but also feel free to go ahead and make sure that throughout the duration of this classroom, please utilize the chat function in Zoom to ask any questions that you might have. John's pretty good about checking it, but also Rui and myself will be checking it, and we can interrupt John at any point if there are any questions or anything's unclear. So we very much encourage everyone to take a look at that. And lastly, before I hand it along, we just want to say thank you so much to John. You know, he's been so wonderful in helping us out with this, and even before all of this, very wonderful and very helpful and during. So we're indebted to you, and looking forward to this class. With that, I will pass it along. I think you are muted, John, so let me...
I am muted.
There you go.
I was just before this meeting, I was reading a news story about how the Senate has been having, and the House of Representatives, and having trouble because everyone can't figure out when they're muted and unmuted.
Sounds about right.
Yeah. I can't find the button. Where's my grandkids to tell me where it is? And then I immediately did to start this one. So, hi everybody. Nice to see you again. John Quinn, Director of Product Development and Science at FlowJo. My email is john.flowjo@bd.com. You can reach me for anything. Tech support is just flowjo@bd.com. You can reach them likely quicker than me, as I'll frequently be doing something like this. So thank you to everyone who replied to our quick little survey poll that came out a couple days ago, which was just asking what topics are people particularly interested in talking about. The first time we did this, we tried to cover just a little bit of everything. And so I thought this time it'd be really nice to hone in on the things that people were particularly interested in. And so let's do that. The winners in the survey of what people would like to hear about were basically the dimensionality reduction tools and the clustering tools, and perhaps comparing what the differences between them are when you might use one versus the other. And so I will focus on that. FlowSOM, actually, as an independent tool, was, I think, the second highest vote receiver. So I'll make sure we talk in some depth about FlowSOM as well, particularly, but I'll touch on all of them, I think, or at least almost all of them. There's a handful that I'll just say, this one's man. So that's that. All right, so let me turn off my video and share my screen. Okay. And so at the moment, you should all see my screen. I just have FlowJo with a little bit of data loaded into it. The first place I'm going to take you, though, is from the Workspace tab to Plugins, and then to the FlowJo Exchange. So just to remind, this is where you can get any of the tools that I use that are in plugin form. So scrolling down, since we're talking about FlowJo and dimensionality reduction and clustering, I can go in here and turn off everything but dimensionality and clustering, reduction and clustering, and get to just the ones that deal with what's pertinent for the time being. Okay. The other thing that I want to point out, so that was really great of Kathy to show a bunch of papers up on the screen that are good reading. Yeah. Some between really good reading, some between really tough reading, but I'll let you decide which is which. And you can get those from the website as well. So if you were interested in FlowSOM, for example, click on it. And under citation, you can get to the FlowSOM paper itself describing how it works. And so most of those tools, most of those papers, if you're interested in the tool, are available just from one click at the spot where you might be going to download the tool anyway. All right, closing that up. Okay, so I think it's worth a little discussion on what the differences and methods are of each of these tools. So let's start with dimensionality reduction. Okay, t-SNE does not appear here. It is in FlowJo natively at the moment. So I will point in the general direction of f-SNE as the representative t-SNE. And just mention that it was the first of these kind of tools that became popular for using in flow cytometry data. Principal component analysis, PCA, is probably the most famous throughout the world, not speaking flow cytometry specifically, but the most famous throughout the world in that it produces a reduced, a two-dimensional representation of your data that tries to maximize just differences, specifically variance between your data points. And so that's used in a lot of places to summarize data. It wasn't particularly useful in flow cytometry because it requires the data to be linearly separable. And so it actually took an iterative approach that is going to just try to map any kind of data, whether it's linearly separable or not, down from a high D space to a low D space. So what t-SNE does is it just figures out which cells are near each other. It calculates the nearest neighbors to every cell, and then it attempts to make a 2D version of that where we just make sure that the cells that are near each other are still near each other. Calculates the distances, pushes them together, measures the goodness of the fit in the low D space compared to the high D space using Kullback-Leibler divergence, and then tries to improve that by pushing similar cells together and then moving apart dissimilar clusters of cells in a second step. And so that's what t-SNE does. It's basically looking to make the nicest picture it can possibly make. Okay, so then UMAP was the first alternative to t-SNE that works in a fairly similar means, in that the UMAP stands for Uniform Manifold Approximation. So a manifold is just an estimated shape, well, a shape within a mathematical space, a high dimensional space frequently. And UMAP attempts to estimate that shape and then maps the shape down to a low dimensional space. And so it's very similar to t-SNE in that there's still some kind of high D approximation, and we try to make that mapping happen in low dimensions. Where it differs is that it attempts to take the distance between nearest neighbors and scale that so that it preserves the distance between nearest neighbors from the high D space to the low D space. And so this will produce perhaps not necessarily as nice looking a graphic as t-SNE does, but it will try to make the distance meaningful from one cluster to the next to say these are more similar or less similar. From high end, you can approximate that in the 2D space. So that's the big difference there between those two. So you would choose to use UMAP if you wanted to make sure your picture, your dimensionality reduced picture, was emphasizing how similar or dissimilar clusters are. You would use t-SNE if you wanted to probably get the nicest looking picture, just to show how many populations are present. And then you might use other metrics to show how similar or different they were. And that might be fine. That might be the point where you say, so we know T cells and B cells are different kinds of cells. They're shown on this plot. One is blue and one is green. And then in other metrics, in the phis of specific parameters, et cetera, counts, whatever it might be, percentage versus parent, there are some differences between my various biological differences, treated, untreated, healthy disease, stimulated, unstimulated, whatever you're looking into. Okay. The third major algorithm that people are beginning to use is one called TriMap. And so TriMap does pretty much the exact same thing UMAP does, up till it gets to the point where it is attempting to scale the data. Whereas UMAP will take your distances between nearest neighbors and say, let's try to preserve that distance between nearest neighbors. UMAP will take points in sets of 3, hence the try and TriMap, and use it to triangulate the distance between them to try to make a better fit compared to what UMAP does, of a nice representation of how similar various clusters are. So this one, you would use TriMap if you felt it did a better job compared to UMAP and were interested in preserving the distances. So it's basically at this point t-SNE versus either UMAP or TriMap, depending on if you want to preserve those distances. And then it's up to you whether you think UMAP or TriMap are better. The jury is definitely still out on which one is better. We've begun to start using TriMap a little bit more in here, and it does seem like perhaps it makes some nicer graphics. Still to be decided. And then the last one is EmbedSOM. And EmbedSOM is based on the self-organizing map approach, SOM, in which you can create a self-organizing map of your data. And we do this using FlowSOM, a clustering algorithm. So I'll talk about that one when we talk about the clustering algorithms. And you can say, I've created just sort of a grid of my data. I'll show cells one at a time to this grid and let the grid adjust to match the cell that is closest to the grid, or sorry, the node or the grid point that is closest to the cell in some approximated space. And then once all is done, you've got once you've shown it thousands and thousands and thousands of cells, you've got a grid that has rearranged itself to the shape of the data. You can then use that grid for clustering, but you can also use that grid to preserve the shape of the data and then use it for dimensionality reduction and apply it moving forward. So you would use EmbedSOM if you wanted to make sure that you could take some dimensionality reduced space and use it in this experiment and a future experiment to be done later and another future experiment to be done after that, because you can forward propagate, if you keep the model, how that makes the data, how it represents the data, it'll just fit it into that model. So the downside of that is that the model, again, will be based on whatever the first set of data you built it on will be. And so it will not necessarily make an optimized or best model for any data set further downstream. And I think, as I mentioned even the first time, I think the pictures are fairly representative, that you get better looking dimensionality reduction if you use something besides EmbedSOM, but its main benefit is that you can forward propagate it. So those are your dimensionality reduction tools. And we could, let's see, I could demo them all, or I could just switch to discussing the clustering ones while we're here. Maybe let's do that, and then we'll talk through all of them in FlowJo. So I'm going to switch to clustering. I see nothing in the chat, so I'm assuming those explanations were the right combination of superficial summary, but good enough to depth to know the difference. Fingers crossed. Okay, your clustering algorithms. And so the main ones here that I want to talk about are FlowSOM, Phenograph, and XShift. You've got flow means. You can look at it if you want, but it's, again, it just basically looks at Euclidean distance and tries to, for however many clusters you put in, find cells that are closest to that centroid. So it's a decidedly mediocre one. These three are significantly better. I've described FlowSOM, the self-organizing map principle here, where we will create the map of the data just by using the data and the distance between them, and then clustering the points within the organized map that we've created. And so what's nice about that one, the clustering part of it, is that it'll be really, really fast. The graphic that it produces to represent what your clusters are nice. They're pretty easy to read. It will produce a series of reports that I think are useful in understanding what has happened in the data. The downside, and it's forward extensible, probably one of the primary reasons to use that. The downside is you have to tell it the number of clusters. It doesn't figure that out on its own. So your other two main options are Phenograph and XShift. So each of these will automatically figure out how many populations are present. So Phenograph, that's a big selling point to it. The other things that it does that it's particularly good for is preserving rare populations and also matching phenotype of what the user, what a biologist might find. The theory behind Phenograph is that different types of cells are going to have fairly different measures. It's part of the main theory of clustering, that the numbers should suss themselves out. But particularly for Phenograph, that if we look at the density in high dimensional space of how our cells are distributed, there should be points that are particularly dense that we could assume are the center of a type of a phenotype. There should be parts that are particularly not so dense that maybe indicate that there's spaces between, and most especially if the distance between a group of cells is very small, so it's a very tight, compact cluster, and there's lots of neighbors there within a very small proximity. So you've got lots of nearest neighbors, then that's a pretty good indicator that a population is real. And so what Phenograph's going to do is it's going to calculate the density for every one of the cells in your data set, and it's going to find out the places where there is a maxima and say, so that's a centroid. And then it's going to go and look for the spots where you've got a whole bunch of cells that are nearest neighbors and then a big gap between another set of cells that are nearest neighbors and separate them off into different populations. And so it's really using how many neighbors do you have as an estimate of, is this the edge of a population or not? And so that's one approach. XShift is very similar in that we'll still also calculate the density of each data point, and then it'll look for maximums, local maximums, and say each one of those is a cluster centroid. And then it's going to go and look between each of those maximums that it calls a unique cluster and make sure there's a minimum, make sure there's a dip in density somewhere. And if it doesn't find one, then it'll merge two centroids together. So Phenograph is going to rely a little bit more on calling cells neighbors and then trying to preserve those neighborhoods. XShift will rely a little bit more on density and making sure that it makes sense that there is high density areas where we're calling things a cluster and low density areas between them to know that they really should be separate clusters and not just a small fluctuation in a very large overall dense population. And both of them are going to then use those density numbers to figure out how many clusters are present without you having to enter a cluster number. So I think that's a pretty big deal for those. I think people have had a lot of success with Phenograph matching manual gating. Manual gating is the gold standard, so that's a selling point for that. And XShift is perhaps a little newer, at least to the exchange, and so maybe it hasn't been used as much, but I think it also does a really nice job, and it's quite a bit faster than Phenograph, which is also in itself a selling point. Okay, so those are the main ones we want to talk about. ICELR has a pipeline that includes some of it as well, but I don't really want to focus on that. And there's a K-finder tool to help estimate K number of clusters, mostly better for SeqGeek and for single cell sequencing data at this point. So moving into FlowJo. Oops, so there's a result we'll talk about in a little bit. What's that for now? Okay, so I just have this one workspace up here, and I've added in a handful of files that have different stimulation conditions. And just the very first thing that I wanted to show, I talked about it, but I didn't show a lot, was the difference between doing some kind of cleanup gating or not. So I just took one of these samples. So we've got different stim conditions. Unstim, stimulation with the peptide that isn't going to cause a whole lot of cytokine action and so forth, and one that should produce a whole lot more of a reaction. So I took the one that we should see a lot of action out of, one of the PMAs, and I just clustered it. I just threw XShift at everything. And then as a comparison, I did some cleanup, and I got down to one of the target population, just CD4 cells, and I clustered that. And then just to show the difference, let's go to the layout editor. And you can argue that it's maybe not totally a fair comparison, but I think it's a good approach. If we had just started at the top and said, tell me what's in this data, you get a whole bunch of clusters. And so here's the clustering. Each one of the different colors is a Phenograph, sorry, an XShift cluster. and it's being shown on a t-SNE plot. And what we see is that this looks reasonable, in my opinion. Again, feel free to always argue, but the t-SNE plot is showing that there are these main populations, and the X-shift algorithm, which was not shown the t-SNE parameters, generally finds what look like the islands in t-SNE as their own separate populations. There's one down here that's red, gray, and blue that X-shift thought was maybe there was more complexity in there than the t-SNE algorithm is able to find, but overall it looks like a fairly reasonable clustering result. In this next plot here, I've used the color now to indicate different populations I did just by hand gating, just in rough hand gating terms. So again, looks fine, doesn't seem obviously terrible, but the first thing I want to point out is I made this neon green population the knot of my singlets gate. And just notice how those neon green cells are spread through pretty much every one of these clusters in a variety. And so just to remind everybody that doublets can look like any kind of cell and sneak into your data and really, perhaps confound you. So it's, it's really worth taking out those doublets. Then this blue population, this one here, are my not T cells. So if the study was really just about, how different T cell populations are affected by stimulation, this is, a fairly large amount of my space, of the space that I can portray my data in, being dedicated to cells that I'm going to immediately ignore. And so it might be worth focusing on a particular phenotype. Again, the clustering algorithm can find them, it separates them. It did a pretty nice job of that, in fact. But if my goal was just to take out and not care about things that weren't at least CD3 positive, then that was just kind of a wasted space that hurts my ability to differentiate the rest of these populations, because they have to be compressed to represent this on our scale. Pink, double negatives for 4 and 8. And again, if we look at some of these populations and say, oh, there's some weird populations in there, I wonder what's this little node in the middle? How come the end of this might be a different cluster than the rest of that light blue one? So right here, double negatives, probably a weird kind of cell that it's going to keep factoring in. So just to say that there's, there are some things that you'll know as an expert that you will say, well, I know I don't want to factor that in, and that can help overall your clustering results. But then if we look at the rest of it, we'll see that there's some good concordance between what we'd expect to find and what the clustering result has pumped out. So alternatively, we could come down here, we could clean up, get rid of the doublets, get rid of the CD3 negative cells, and get rid of the 4-8 double positives, and show those data on a t-SNE plot. It's going to end up looking fairly similar, but a lot more, say, compact. And then we'll notice things like, okay, so it looks like the structures that the t-SNE plot found are match very closely with the clustering algorithm is showing, that there is some population in here that perhaps X-shift thinks maybe has a little more complexity to it, particularly that gray end does seem to be separating as it is, that if we look at my manual comparison that I said, well, those are IL-2 positive CD4s, maybe there's something more going on here. Maybe the combination of parameters indicate that that is a different cell type. And maybe there is some subset here of these tan ones that are worth it looking at. And then we can see also the things that I called CD4 just TNF positive, which seem to stretch into a second population here that maybe I didn't consider when I was gating, do get separated by Phenograph. So again, just a handful of pictures to say cleanup was worthwhile, and this is how one might see those differences manifested when looking at the data. Okay. All right. In terms of comparing the different plots and how to use them, so just off the bat, that was the PMAs for some stimulation with t-SNE being shown. If we were going to run any of the clustering tools, and maybe let's just do it on those CD4 positives that are not, clustering, of dimensionality reduction tools that are not t-SNE, we could go to plugins and we could go find either UMAP or TriMap in here, or embed SOM for that matter. Click it. you will get some kind of parameter selection. It'll generally highlight everything to begin with. We can go pick, and actually, let's take it back up. Yeah, okay, this is fine. This is fine since I did set up that one, and that'll make an easy comparison of one list that we actually have to run. I'll highlight the parameters I'm interested in. Distance functions, Euclidean, Manhattan, Angular, and Hamming. Basically different ways that you can approximate distance in a high dimensional space. Like when you have, say, 4 dimensions, what's the straight line between 4 dimensions? Well, you have to specify how you're going to do it. Euclidean is definitely the most common and the one I will pick. And I overall don't know that that the data are particularly sensitive to the different distance metrics. It seems like a reasonable choice. Nearest neighbors in this case is, if you remember, both TriMap and UMAP are going to try to figure out basically like local density. And part of that is saying, well, how many nearest neighbors do we want to assume matter? And so again, we found that the data are the results are relatively robust against changing these over a fairly broad spectrum of numbers. And we're going to stick with the defaults and say go. And then UMAP will take, or sorry, TriMap will take a couple minutes to calculate. We will see 2 more parameters appear underneath these populations. So up here we've got the t-SNE of CD4 and the t-SNE of PMA, just indicating that one was run on the total population and one was run on just the CD4 population. And then we can show those 2 maps next to each other and compare and contrast. So what I think we'll expect to see is that, again, that the t-SNE I think will look a little bit nicer, and perhaps the TriMap will maybe point out some smaller populations that pop out of it. So calculating.
And as that's calculating, and just a quick reminder to everyone, if they have any questions or anything's unclear as we're moving along, please feel free to use the chat function.
Awesome, good reminder. And while that's calculating, probably time for just a quick mention of the plugin wizard. So you'll have to be clicked on some population, doesn't matter which one. Plugins and plugin wizard right here. Also available on the Exchange. Just, oops, and Xshift is calculating again too. We don't want that to happen. A lot of times if you run a clustering algorithm and then change something, it will assume it needs to rerun. Here, the only thing it was running was a different dimensionality reduction, so I didn't really want Xshift to recalculate. We'll go back and stop that in a sec. But plugin wizard is just a tool to help you get these plugins up and running. It makes sure that you've got a path set to where FlowJo can find R. It gives you a place to go get R if there's any of the R-dependent ones that you'd like to run. Amongst the tools that I mentioned, let's see, I think only, so Xshift is just written in all Java, so that doesn't require R. Phenograph does, but it's sort of a proxy for, it was originally like a Python tool. FlowSOM does. So amongst the clustering algorithms, about half of them do, I guess. And then amongst the dimensionality reduction tools, t-SNE doesn't, it's built in. UMAP and TriMap do. All right, anyway, so plugin wizard, connection with R if you want. You can check this box to have plugin wizard auto install the various R packages for every plugin, a useful tool rather than having to do them one at a time. You can set the path to where your plugins are, although again, generally you will have already done that to be able to have run the plugin wizard. The what's new in the Exchange button will show you the version you have installed and the current version of all the plugins, and give you a chance to, if you click on any of the buttons for latest version, go immediately download them. And advanced has places where if you wanted to run Phenograph generally, you'll want to have exports and just a copy of what's on the Exchange and a way to get them all. All right, don't do that now. Stopping Xshift. TriMap should finish up in a moment. I assume I slowed it down by resetting all of the other calculations going on. All right, while that is still calculating. The other thing to mention is that all of these clustering and dimensionality reduction tools are going to create fairly novel new sets of parameters or novel sets of cluster numbers. It is worth combining all of the data that you intend to do some work on. And there we go. Thank you, Plugin Wizard. And doing so via concatenate. Oops. So let's just make, sorry, scroll bar. And where is the TriMap version? Here we are. Oh, I was up a level. So anyway, here is the TriMap of X and Y. Okay, and let's grab that in the layout editor for doing some overlaying. I will come back to concatenate in a second since this finished up. Okay, CD4s, drag them in. Option clicking to make a copy. And then we can show the t-SNE of the CD4s right next to it. t-SNE of CD4. Okay, so yeah, fairly nicer, more spread picture if you want of the t-SNE, perhaps some better separation. And then if we want to say take some of these CD4 populations that I just hand gated and overlay them, you see again the plot that I showed before, without quite as nice a color scheme in use. When I now choose two very similar colors, let's make one of those green. Okay, and drag those onto this plot. And make sure colors are relatively the same. Cool. Okay, so and again, this was just the hand gating as opposed to the clustering approach. So likely my hand gating of TNF positives could have gone another segment deeper. Looks like there's two distinct populations there. But again, the clear differences between t-SNE and UMAP, these populations are really well separated. They're far apart. Doesn't look as pretty, and so it's up to you what your preference is on how you'd like to do that. And you will tend to see a little bit more of that, like straight line. I think appearance in both TriMap and UMAP. So that is those two. We could also run UMAP. Maybe we'll do it on the concatenated version of a data set and give it a little more time to run in the background. So I'll mention we can come in here and we can try some of the different clustering algorithms and view them in t-SNE and UMAP. I also have another workspace already put together that has, I just downsampled the concatenation of all those files. So I should come back to how to do that, and already built a t-SNE on it so that we don't have to wait for that to happen. And we can use that as a way to look at how the different clustering results work. We could also maybe run UMAP on this in the background while I talk about concatenating and some of what the clustering results are. So let's do that. Let's hit UMAP. So we'll have seen one of everything. Just pick out all of the fully stained parameters. UMAP, similar, has a set of different distance measures you can work with. I tend to pick Euclidean. It's got nearest neighbors settings, and we can say go and let that run in the background. While that does its thing, I did mean to mention how to concatenate. So with the concatenation, if I wanted to do any cleanup gating, we could say take our singlets, maybe T cells, drag that to the group so that we have a cleaned up version of each of our parameters. Select the T cells. I like to use the Edit Select Equivalent Nodes tool just to immediately highlight all the T cell populations. Right click on that, export/concatenate, choose concatenate, Format FCS3 sounds good. Desktop as a location, fine. The parameters that you'd like to export, generally all. So it can either be all uncompensated or all compensated. If you have acquired your files and compensated them on the cytometer, so they have a built-in acquisition matrix, all uncomped is a nice choice because you will get the raw data and the comp matrix all exported together, since the comp matrix is stored as a keyword. So you'll get uncompensated data and a keyword for the compensation matrix. All compensated is nice if you have done the compensation in FlowJo, or you are combining files that have different compensation matrices, perhaps time point 1, time point 2 of some experiment, et cetera, and you want to put it together. Custom would allow you to pick and choose what you want. If we would look here, by default, you're just going to get all the parameters, but we may have wanted to leave out some of, say, the clustering results, possibly. You'd probably want to leave in the t-SNEs if you bothered to do them, maybe, but we might be exporting this to do t-SNE uniformly on all of them, so if you've run it here, you may want to just leave that out also. Anyway, down here, I will point out that you can add an extra parameter as a keyword. I think when I did that to create the workspace in the background, I just added a 1 to 9 for the 9 files, and you'll see that pop up as an extra parameter, so that you can separate out the data once you have concatenated it all together, and then do some comparison between the various files, and then just hit concatenate. You can just do this. Oh, sorry, if you want to rename it something, you could type that something here as opposed to concat. Good thing to do since I've already concatenated and have a file sitting on my desktop called concat. And then you can either add it directly to a new workspace, to the existing workspace, or just close. And I'll now minimize this guy. And that was part of that one, so we don't need that guy up. Okay, and then so over here, so UMAP is still calculating there, that is cool. If I double click on the concatenated file that I added to this other workspace, you can see that there is just a parameter called number. And let's just put that back on a linear scale. We can probably tighten it up a little tiny bit. Okay, and those are each of the 9 files, just in order of 1 to 9, and we multiply by 1,000 to put things on a flow cytometry scale. If you show this as a histogram and right-click on it, hit gate on peaks, you can break these back up as our individual files, so that at any point we'll be able to see what the unique contribution of any one file is. We'll expect some differences in this data, as some are completely unstimulated, some are stimulated just a tiny bit, and some a lot. So if we would look at something like interferon gamma, we'll expect to see some difference in the expression levels, particularly the middle 3 there. The PMA stimulation way more powerful. That's kind of what we're going to expect. IL-2, again, a lot more in the PMA, the 3 PMA samples in the middle. So we'll expect to see some difference in the populations that emerge out of the various tools. All right, and we can do some comparison using t-SNE to represent the outcome of our various clustering approaches. And we can also, once UMAP wraps up, show them on UMAP. So let's see, UMAP should be finishing soon. I did feed it, what, 2 million some cells, so that's a slightly longer process there. Well, while that finishes, let me also point out that I think a nice tool is the color map axis. If you're looking at t-SNE plots, it'll show you positivity for a particular parameter. We took CD3 cells, so they're all fairly positive for CD3. But if I looked at some other parameters, CD8, we'll see that we've got a few different CD8 islands. Probably these are mostly the CD4s, and there's some double negatives in there, we'd assume. So there we go. And so on. And that the layout editor can be used for seeing all of these things at once. Just going to show those t-SNE parameters. Right-click on the plot and say, make a color map multigraph overlay, and pick the parameters that I'm interested in, which is probably all of them except for CD3. Okay, so there's our profile of the data. Oh, and I could have probably also shown CD8 in that. Oh, and it was there. Whoops. I was thinking I selected it. Okay, I was IL2, I forgot. Okay. Anyway, yeah, so 4s, 8s, pretty much everything for 3. TNF across the top, interferon gamma over here. IL2 spread throughout, bunch of double negatives, and a lot of double negatives in this particular data set, since there is a a fairly large bit of unstimulated files and then minimally stimulated. Question from Sarah, is there a way to get the parameter name in the multigraph overlay? So that was, what was it, 10.6, I believe, came out with the omission where we only showed the PNN, the parameter name, so like the PE size 7-bit, and I believe it was 10.6.1 that we added also the PNS, the stain or specificity. So if you have one of the latest versions, then you should also get the parameter names there, the IFN, the CD8, CD4s, etc. All right. UMAP still calculating. I hate to throw a clustering algorithm while still thinking about UMAP. My apologies. I thought to run the t-SNE on the bigger file before we started. I should have done the same with UMAP so that we could have actually looked at these without having to wait for it to finish calculating. Anyway, sorry. But what my intention here is to do is just to take this downsampled population, and it is downsampled because I know I typically say I don't like to downsample. I like to keep all of my populations in there. The one time I will downsample is when doing demos in front of a live audience, and it's already taking this long to do the calculation using UMAP on the downsampled population, just to prevent people from waiting for too much. All right, come on, UMAP. Well, I did downsample to a much smaller number. It shouldn't be too much longer. Anyway, so we'll take, sorry, mid-thought, the downsampled population, and we'll run FlowSOM on it, look at all the results, Phenograph on it, look at all the results, and XShift on it, and look at all the results and compare. And then just show how to batch out some of those outcomes on the various samples 0 through 8 that we've added to this data set. And so this is also showing one advantage of t-SNE compared to the others, because it's built in, it's a lot faster. And my impatience with running some of the ones that I have to run as plugins is showing. All right, up down sample, just checking, so good. All right, well, maybe we'll attempt to run simultaneously and hope that it'll calculate the quick things like clustering first. All right, so let's throw FlowSOM on there first, since that was the one that we got the most requests for. All right, so again, FlowSOM, self-organizing map. The top portion here is just a selection of parameters. Ooh, I selected to export the uncomped CD3 and the comp version. Well, let's deselect uncomped. Okay, so I've picked the parameters I want to feed it. I have not picked a particularly high dimensional data set here. I want to make sure I found one that had some differences in populations and to try to make this go fast, since a few of these are intensive operations and I did want to concatenate and feed it a lot of cells. So, so anyway, sticking with what, seven dimensional data at this point. Everything we say here in 7-dimensional space applies to 20-dimensional space, just it'll take a little bit longer. The number of metaclusters that you will use to set your FlowSOM final outcome as is an estimate. It's however many if you've made, say, a t-SNE plot, which we have here, and there should be at least, say, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14-ish, 15-ish maybe. Let's say 15. If you haven't made a t-SNE plot, you could think there's 1, 2, 3, 4, 5, 6 parameters we're using. You could have as many as 2 to the 6. That would be your upper bound. There will not likely be every possible combination. You might not care about every possible combination, such as things that are CD3 negative, 8 negative, 4 negative, etc. takes up a lot of those combinations. So that's just some idea though, how many variants of the different parameters that you could be positive or negative for that you would like to run. So anyway, we'll say 15, having glanced at a t-SNE plot. And the other thing we'll do is go and look at some of the outcomes that come out of FlowSOM as a check once we've run it to see if that seemed like a reasonable guess. The SOM grid size, width by height, is just setting the dimensions of the net that we will use, like the grid that we will use to start the self-organizing map. 10 by 10 width by height means 100 nodes used to map the total space. That is usually going to be enough. You can view the layout as a t-SNE plot, as a grid, or as a minimal spanning tree. I've got the t-SNE plot here, and I think FlowJo has a better t-SNE than the FlowSOM algorithm does, so I wouldn't pick that one, and a grid is dull, so I really like the minimal spanning tree. And we'll talk about exactly what that is once we get some results. You can plot the channels as heat map for a given parameter, or all of them as a pie chart. I like that choice. You can ask it to fix the node size so that the nodes are all the exact same size and makes them a little easier to read, or leave that unchecked so the nodes vary in size, and the bigger the node, the more cells that are ascribed to that. I like to leave that unchecked. And I do like to leave save the R script and output methods checked on so that if anything goes wrong, we've got a script of what happened, and you can either look at it yourself and try to fix whatever the problem was, or you can ask our tech support team for some help and see if they are able to then just look at that script and go immediately to what the problem is. So I'm gonna say okay and see what happens here while we're trying to calculate two things at once, while also doing a screen share. So we're being brave.
So John, I have a quick question. How quick these algorithms work, or these plugins work? Does it depend on computing power, and what are there's like minimum suggestions by FlowJo that people need to have for their computers to be able to operate?
Yeah, it's absolutely based on computing power, but probably even more so the size of the data that you hand it. So at the moment, I've given it 500,000 events, 7 dimensions. So, that's probably actually like a middle of the road kind of size data set. t-SNE, I will claim, ran in that in, 8 minutes or so. UMAP's going to be a bit slower because it has to interact with a plugin interface. I imagine FlowSOM would actually run in 30, 40 seconds or something if I wasn't also running UMAP at the same time. So anyway, minimal recommendations really just come down to your RAM. It's processor speed, number of processors, and RAM are the things that are going to matter. Number of processors, that one is, that one's kind of the major price point of which you get when you're buying a computer. 4 to 8 to 16 processors, or cores as they're frequently called, are all like big jump points in price. So you're going to tend to get what you're going to, what you can afford or what your budget has been given there. This is a 2 processor machine that I'm working on right now. People have better, but it's a Mac, so it's got that going for it as well. So processors, the more you can get, the better. There's no minimum requirement there. You can run with 2 processors, that's fine. I don't think you can buy a 1 processor machine anymore. If you get more, it'll be a lot faster. Pretty much processor speeds are the best to the worst. There's not a whole huge difference. They're all in some large number of gigahertz. The place where there's a lot of variety that will make a big difference is in the RAM. The RAM is your computer's easily accessible memory, the stuff that it can think about right now, as opposed to pushing to like a long-term storage. So again, more is more. The minimum requirements are highly dependent on your data set. There really is no minimum requirement. It'll always run. It'll just take much, much longer with less RAM. So the biggest threshold difference is if you can have enough RAM so that your whole data set fits into like the working memory, which is what RAM is, then that'll prevent your computer from having to do a bunch of input-output operations, switching things from local memory, working memory, to long-term memory. And so you can estimate that by, if I went to this data set, right-clicked on the folder, ooh, less screaming. Sorry, a crowd in the background here having trouble apparently with school. Where was my ICS data set? There it is. Oh, sorry, I have to show and finder. ICS dataset and get info. So 343 megabytes. Oh, and that's done. Cool. And so, you know, I'd want to have definitely at least that much RAM multiplied by, you know, 1.5 or something like that, because the computer operating system is going to take up some amount of that memory just to have power on and be showing you your desktop and so forth. So pretty much more is always more. The place where you can make the biggest impact at the smallest price difference is just adding RAM. Frequently when you're computer shopping, to double the RAM adds $100 to the purchase. To go from, you know, like a 4-core machine to an 8-core machine can double the price of the machine. So, you know, that may have a bigger effect, but also you pay for that bigger effect. So sorry for not being able to say that there's an exact number of RAM that you should have and an exact number of cores that are the absolute right number. It's definitely a relative scale based on the amount of data you have and the amount of money that you're willing to spend. So I hope that's a good enough answer. Sound okay?
Yes, thank you.
Cool. Okay. So all that finished at the same time, which implied that UMAP was dominating the memory. And as soon as UMAP was done, FlowSOM ran in a couple of seconds. So good times. Okay. So let's take a look at those two results. Just dragging in downsample, switching to t-SNE 1 versus t-SNE 2. Okay. And we can drop our FlowSOM populations on that. Okay, get something like that. I might double click on that and switch from contours to dots. I like that a bit better. Okay, and some of those colors are iffy. I also will generally, when I'm first looking at my data, see if there are any populations that are just too small to worry about. These are actually all okay. Sometimes if you pick too big a number, you'll end up with a population with 5, 6, 7 cells. The smallest here is 646. I believe that could possibly be population, so let's leave them all. And maybe just adjust a color or two to be a little more different. Oh, that was a very small population. That was the 646, and I just turned it orange. Okay, and maybe let's make this one. Yes?
I'm actually going to ask another question. So speaking of that, with number of cells, is there a minimum threshold that you recommend for a population, and how would you tackle like extremely rare populations that people might be looking for, very like unique, like 1 in a million type cells?
Gotcha. So a lot of that is going to come down to collecting enough events and having an idea of how rare your target population is. It could be that you know, you don't know everything you're looking for, and so small populations will pop out. And at that point, it's up to you to one, decide if you've got enough events in that population that you can get any statistical significance from it. There are just a few equations on if you know how many you're trying to, what the rarity of your population is, how many events you should collect to help achieve a certain number of statistical significance. And the other thing to do is if I didn't know, then I would look through. And if I did have a population that was, you know, something under 50 cells, I would be extremely dubious about it. You'd want to look at it. I'd also maybe choose some clustering algorithms like Phenograph that are particularly good at clustering for rare events. And you know, it's going to attempt to highlight things that have that are very, very tight. It may be only 50 events, but they're all really, really similar. It gets a cluster. If they're not really, really similar, they don't get a cluster. And so you will tend not to get clusters based on tiny, small populations that aren't real. So something like using Phenograph will just help. And then the other thing is if you haven't done it, or you're just trying to look at a population and see what it looks like, it could be that it really, really stands out nicely. And you think, well, that's beautiful. You can look at the population for consistency. You could check something like the standard deviation of any or all the parameters. You could look at the next closest populations and see how similar they are. FlowSOM is very nice for that, in that it puts out a really nice heat map chart. You can create similar things in FlowJo, but FlowSOM will just put it out just as part of your results. And if you look at cluster 1 and cluster 2, and cluster 1 had, you know, a large number of events, and 2 had very, very small events, and you look at their profile across all the different parameters and they're extremely similar, you might think, well, it's enough just to it's enough just to assume that that's part of that main larger population. If you see that there is a parameter that it's different on, then that's worth considering. All right, is this parameter something meaningful that me as the expert on what this data is, that a difference in that measure says something to me, that, that this really could be a very different kind of cell? I think, you know, that's where some of your expertise will really help in trying to decide if the differences you see in some small populations from its nearest neighboring populations are relevant, and where FlowSOM and Phenograph will each have their strengths. Again, Phenograph just not producing small clusters if they don't feel real to it based on the number of nearest neighbors, and FlowSOM in being able to very quickly see what are the closest other similar clusters and what their profile is. Okay. All right, so I was just going to then switch to UMAP to compare these things. And I do think we've got two purples and a purple and a dark blue that are really similar colors. I'm gonna switch in both of them. And just make like a red. Oh, maybe that was mostly all one cluster and they're just kind of overlaid a little bit. Oh, all right, never mind. They are mostly one cluster. Okay, so again, t-SNE, if we're just evaluating the dimensionality reduction tools, tends to push things apart a little farther and spread them out and make it a little nicer to look at. UMAP is telling you how different these things are. So it's telling you that this green, purple, light purpley pink, you know, these are all really, really similar, whereas they're a little bit more spread out here in the t-SNE outcome of this. And these are all just our FlowSOM populations. We could, if we wanted also as a map, just bring in one of these again. And we could heat map by parameters to get a feel for what's where. Again, knowing that things up here tend to be the more interesting, smaller populations that are expressing a variety of the functional markers, 4s, 8s. We could have done the same thing with the UMAP representation of this to get a feel for, but we can see, because I've kept the colors consistent, that these populations are tending to be in here. So again, some of the more interesting populations are made to stand out a little bit more on t-SNE, whereas they kind of crush together on UMAP, indicating that they're different from we're going to assume that this population here, the red, this broad thing, which is the middle of these and which is largely green for everything, that these largely red cells in these two plots are all our negatives. And that and so we can see in UMAP that our negatives are much more different from everything else, and that this blue population stands out over here just because it's the CD8 version. And it looks like a lot of, there's some CD8s in here. Anyway, it's just a big, CD8 is largely negatives, and that's why they stand out as different from the rest, up this way. So again, each of the different types of tools here is telling you something different. Combined, actually, it's nice to kind of get a holistic view. And there's our FlowSOM populations. Now let's check out the folder of outcomes that FlowSOM produced. And actually, while we are doing that, I am going to run Phenograph so that we can compare the results of the two clustering algorithms also. And just turning on Phenograph using the default nearest neighbors. Okay, so this is Friday Flow 2. Opening up the Friday Flow 2 folder. It was automatically created when I saved the workspace and ran a plugin. I've got the outcome of my various plugins that I've run in here. Let's look at the FlowSOM outcomes first. Okay, here we go. Up top, we've got all the different pictures. So a PDF and a PNG version of each one of them, and then a CL for cluster and a POP version for each of the heat maps and the labels, and then a table of MFIs. All right, looks like Phenograph maybe has done its thing. Cool. Okay, let's finish looking at this first. And so the POP heat map, if we open that up, is going to show us a dendrogram of each of the populations. So that lets us know that populations 6 and 11 are the two populations that are most similar to each other. And so this is the bit where where we can assess, are these things really different? What if one of these was a particularly small cluster? And let's go for real on that and say, well, we know that, population 10 was a particularly small cluster. So we can go find 10 in the list. You can see that 10 and 9 were the most similar populations to each other. And then those two actually, by the length of the line here, were fairly different. But overall, those two are pretty similar to the next most similar to 1 and 12. So we look between 9 and 10, and we see actually that they are, CD3 mid-bright, CD8 mid-bright. So these are our CD8 T cells. And one expressed TNF and one expressed IL-2, and they didn't express each other. And they were both lacking interferon gamma and, well, of course, CD4. So I immediately say, okay, those are, that to me, biologically, that's relevant. That is two different functional markers have been excited. Those are two flavors of T cells that are distinct. And even if one of those populations is very small, then I believe it's a real cluster, and I'm going to keep up with it. And if we glance at the next two up, we also saw that these are 3s and 8s. And what they are is the interferon gamma positives. However, one of them is actually probably negative for 8, but it's one of our double negative populations. So we're a little suspicious about population 12, which also was a fairly small population, 0.55%. So yeah, we're wondering about 12 biologically, and that perhaps as it's a double negative, I'm not sure how it fits into the T cell landscape, being naive. From Sarah, what troubleshooting can you do if dimension reduction algorithms do not separate well geographically? I saw this recently in mass cytometry with 20 plus surface markers and 500,000 plus events in the file. So separate well geographically, let me think, what would we mean by that? That you tend to get more of just a tight pack of cells in the center of the screen, for example, as opposed to well separated islands. That's my interpretation of separate well geographically as opposed to phenotypically, whereas the things that we could say manually gate and compare look like they're well separated. Is that about right, Sarah? Yes, awesome. So the most common issue there is that most of these most of the dimension interaction tools have some kind of goodness metric that they're checking on in an iterative manner. And depending on how much control you have over your algorithm, you can stop too early. You can use one phase of the algorithm too long. And a lot of people have pointed out to the number of iterations in t-SNE particularly, and when the early exaggeration or the first portion of the program switches to the second, which is pushing apart dissimilar clusters. And so the answer for the general answer is you should ask for better control of your tools. With mass cytometry specifically, people have tended to use some of the cytobank tools for that. And Annabelle Kina, who wrote the opt-SNE paper with us, was one of the original, one of the real pioneers of using this technique in cytometry and with some mass cytometry data. And when she wanted to use it in cytobank, she actually had to get them build her a special set of control panels so that she could actually tune those to exactly what she wanted it to be. And her frustration with having to tune this time and time again was part of what got her to do opt-SNE. So the FlowJo specific answer to that is that I just highly recommend using opt-SNE. The tool Anna created that we helped her build in. And so if you click on some population, whatevs, and go t-SNE, there's this learning configuration auto for opt-SNE with the reference right here. Is just a means of having the algorithm automatically measuring what the goodness metric is while running and making decisions on when to stop a phase and start the next phase based on actual measures in the data. It's just a much, much better approach. And we shouldn't see just the ball of death, I think is the most common expression for when your t-SNE just comes up as one big ball in the middle of the screen. We should do a better job by automating the process of when to move from one stage to the next. We don't have those same level of controls built into UMAP or built into TriMap. Perhaps they don't need as much levels control, as I don't think they have the two distinct phases. They will try to, again, estimate a manifold, so that if you don't get a good result out of UMAP, it's likely the amount of runtime it was given to optimize. And that one is one we have less control over. We'd spend an awful lot of time building very precise controls into t-SNE and building in algorithms. Another reason why I like that one specifically. Okay. So back to the heat map. Again, just gives you an idea of what your clusters are quickly, and trying to differentiate why one might be separate from the next, if any tend to be part of the same cluster, etc. So these all look pretty well separated between almost any pair of clusters we see, and they're lined up to be next most similar. We can always find at least one parameter where they are very, very different. So I think 15 was at least the number that we wanted. If we wanted to estimate, should we have had more? We could go into that Friday Flow folder again, FlowSOM, go look at the clusters, heat map. So this is the 100 nodes. This is plotting each and every one of the nodes that we started with that were then clustered. And overall, this is a pretty tough graph to look at. I don't know if you're going to draw major conclusions from it, but what you can do is just look at how the dendrograms line up and see how many clusters we see naturally. So this is all perhaps one set of clusters that are all very different from everything else. There's a big long line here, and perhaps within them, well, this one looks like it was perhaps an outlier. It by itself, there's one row that's very different from everything else, but maybe there's one more group here that is distinct from the others. So maybe that's two and an outlier perhaps. Perhaps this is 3, 4, 5, 6, 7, 8, 9, something like that, but we could probably decide that there is a couple, maybe these are too distinct. Maybe we should have 10. All these lines are pretty small once you get past separating out those two. And then maybe this is, one more outlier, and depending on how many clusters you want to break this into, maybe there was actually 3 or 4 there. So 15 was actually probably a pretty good number. That's done too. So we could have maybe gone up to 16 if we said we think these are each 1, 2, 3, 4, 5, 6 right at the beginning, 7, 8, 9, I'd say 10, 11, 12, 13, 14, 15, and then depending on if we want to go, I guess it would be 16 or 17 if we wanted to give the outlier clusters each their own node. But anyway, it's a way to certainly get within the ballpark. I'm thinking very clearly that the number is between, say, 14 and 17 clusters that we wanted to run. And we could rerun FlowSOM if we want to try it with a slightly different number and see what happens. But that's what that tool is for. The labels tool, the labels graph that you get here is just when we look at the FlowSOM plot, if we wanted to see which population is which, we can. There's 2, 3, 4, 5, and that I didn't even show yet is actually just the icon that FlowSOM creates. So in addition to looking at these on t-SNE plots or UMAP plots, we could also, let me just hit the plus here, look at the FlowSOM minimal spanning tree. Okay, so amongst the files that we get, a bunch of heat mapping and so forth, and then a little explanation of this. What this now is we asked it for 15 clusters. Those clusters are identified by the colors. So various reds through pinks here with greens and teals. Some of these, as you get to larger numbers, gets very difficult to see the difference between, say, 4, 5, and 6. And that's maybe why you wanted to have brought up that plot that identified exactly, which one from the outputs here. Pop labels. Sorry, attempting to double click. So 4 was here. 5 is actually an outlier. 6 is in here. 6 is with a really small one. I remember that now. Anyway, so those are the 3 greens and which is which. So 4, 5, 6. And then each one of the circles is one of the 100 initial nodes that are then the color overlaid on them is the indication of the clustering. So clearly this pink cluster is the biggest, then comes the red, a few different outlier clusters, a few smaller clusters, and then we can use this legend over here to figure out what each of the clusters are. So that green one we were looking at, the colored pi wedge, the closer it gets to the edge is how positive it was for a particular parameter. So that's interferon gamma, IL-2 expressing, CD4 positive T cells with a little bit of TNF. So these are highly polyfunctional T cells here. And we can see that maybe here's the 8 version of that. And just more TNF in this population here. The pink ones here look like they have the brown and the blue wedge. So these are wedge pretty extreme and nothing else. So these are just CD4 T cells that didn't have a lot of the cytokine activity happening. So probably coming from the unstimulated samples, which there were 3 of. Here's this red population here is light blue for CD8 positive. CD8 positive, CD3s. So there's our 8s that weren't doing a whole lot, are the two main populations. Again, making sense since 3 of the files were totally unstimulated, 3 of them were minimally stimulated. And I bet what we would see was that a lot of these more edge populations came from our PMA stimulated files, which we could check. Okay, so anyway, that is the outcome out of FlowSOM. And again, I should have mentioned that the line between each one of these nodes is indicating how dissimilar it was from the next node. And so these two are actually fairly dissimilar, lumped together because they're phenotypically very, very similar. Other than, there must be some difference, looks like in a tiny bit of TNF. All these pink ones are extremely similar, these nodes, hence they're all combined into one cluster, obviously. And some of these like purple ones get outliers because they're very dissimilar from everything. All right. Let's also look at the difference now between using a different clustering algorithm. Oops. Okay, so we've now done, we can grab the same plot if we want and just show the Phenograph results. I'll move this guy over. I'm going to hit, actually, let's not do that. Let's just bring in the downsample one and I'll just overlay the Phenograph results there. We can do t-SNE and UMAP if we want. And then Phenograph will actually, I should point out, figure out how many clusters there are. Automatically, it looks like it said 29. All right, so there's our Phenograph results, but really broke down a lot of these populations even finer. Of note, we've seen in all these plots, there is this little population that stands out right here that even, is just visible throughout, that is just one population of cells that were like particularly negative for interferon gamma, just a little different than everything else. Some kind of odd variant that so far only Phenograph, I think, has picked up as its own population. So that's kind of neat. If anything, perhaps it overclusters a little bit, and we may want to actually have gone back and we might want to go back and compare some of these, but it's certainly not going to miss anything. And then we could also duplicate this plot, just holding down Option, and show these on the UMAP. On the UMAP scale, and give you a feel for that. And so that actually looks on the UMAP scale. That's pretty interesting, isn't it? That the Phenograph populations are really nicely, tightly lumped together, again, indicating that perhaps UMAP is doing a bit of a more accurate job, even if it doesn't look particularly nice. That everything Phenograph said was a cluster just sticks together, as opposed to being a little bit more intermixed and spread out. So here's the option with those two. This again, Phenograph, particularly good for trying to find rare cell populations. And we can see that if anything, it overdoes finding rare cell populations. It's definitely picked out a few things that I think obviously stand out to the eye as different, that were maybe missed by FlowSOM. FlowSOM, a few more tools to dive into your data, and was definitely quicker. So why don't we also do X-shift on this data while we're in here, plugins and X-shift. X-shift is a bit faster than Phenograph, and so I think we can expect this to complete in a reasonable amount of time. Again, the defaults are generally fairly good. If you want to take a deep dive into them, the papers are present for all of these on the Exchange. We've tried to give you the full set of controls, but we've tested a large range of defaults, and it doesn't seem to affect the outcome too precipitously in any direction. Okay, and I think X-shift is one that'll just run in just a couple of minutes. How are we doing time-wise? Time-wise, we are almost at the end here. So I think this will be the last thing we do. And then perhaps I will quickly just again mention that Cluster Explorer is a fairly nice tool for exploring any of these, and you can use any of the dimensionality reduction tools or clustering tools with it. and switch between if you'd like. And just a reminder too, again, X-Shift and Phenograph, that one's done. Both k-nearest neighbors density estimation based. The second pass of how it figures out the clusters is where they vary, whereas Phenograph is looking to make sure that you've got populations with lots of neighbors by default, and X-Shift is making sure that you've got local minima separating your maximums, your centroids. Moving that one farther over, dragging in downsample. Let's do t-SNE at the top. And X-Shift thought there were, expand that a little bit, 15 clusters. So that seems a bit more reasonable than Phenograph's 20-something, 28 or 29. In fact, that looks a bit better, but some of these I would think maybe should have been more than one cluster. Let me just make sure the coloring isn't just set so that we're thinking that it's one cluster, but actually more than one thing. Yeah, that was the case there. Okay, but that one, yeah, that one does feel like that should be more than one cluster. So let's option this down and let's show them on UMAP. Well, that was t-SNE versus t-SNE. There we go, UMAP 2 and UMAP 1. Okay, and again, showing a lot more homology here. And again, combining a whole bunch of these here, which were separated by Phenograph, by FlowSOM. So anyway, there's your comparison of the 3 different clustering algorithm approaches. UMAP will definitely go and dive the deepest and take the smallest populations. X-Shift looks like it's done automatically a number of populations as a reasonable job. I'm going to subcluster some of these. FlowSOM required me to tell it how many there were, but overall I might like the result better. And I find myself over and over again coming back to FlowSOM as my favorite clustering algorithm. I think perhaps for good reason. So anyway, very quickly, let me just show you one more tool. I've probably shown it once before, maybe in the last one, but I like it a lot. I'm going to go to Plugins and Cluster Explorer and just point out that since I've run Phenograph, FlowSOM, and X-Shift, I've got 3 different choices of which kind of, which clustering result I want to show. We could maybe do those X-Shift ones. I can look at which parameters might be interested to plot. I'm going to take out the scatters, just leave these guys here, and sample ID and sample number. And then I can choose which visualization technique I want, my t-SNE or my UMAP. I'll say the t-SNE, say OK. And then Cluster Explorer will build me an interactive viewer for going through my clusters. Okay, so there's my t-SNE plot. Here's my list of all my clusters. Here's very similar to FlowSOM heat map version of them, so I can try to decide why and how they're different. The line plot that shows where they vary the most. So, oops, included the t-SNE parameters in there. Should have taken that out because, of course, they're very, very different on t-SNE parameters. Maybe a lot of variety on CD4, on CD8. We've got strongly positive and negative. One population that stands out, the blue one, as T and F particularly. And then we can click through and isolate by population what they are positive for, or compare one or more by highlighting them. Okay, with that, let me just check back at the chat and see if there's any more questions. So hopefully that was a fairly deep comparison of the main clustering and dimensionality reduction tools, what they're doing under the covers, how you might use them, how you might compare the results, and hopefully some indicator of which one might be best for a particular use case.
Okay, great. Thank you, John. That was wonderful. Definitely very comprehensive. Does anyone else have any questions before we wrap up? I think we're looking good. Again, I just wanted to say thank you so much to John Quinn for being so generous with his time and helping us out with this today and for our last session. This was recorded, so we'll be sending out the recording once it's uploaded to YouTube and then And then after that, our website, probably by tomorrow or maybe I'm ambitious tonight. So we want to say thank you to John. Thank you for all of our attendees. And this isn't the end. We'll continue to work on more virtual classrooms, not just in FlowJo or FCS Express, but also just general virtual classrooms in flow cytometry. And we'll be communicating with that, or as we progress with that in the very near future. So just be on the lookout for some emails from us. And I think that's it.
Awesome. Thank you for organizing, Kathy. Greatly appreciate it. And maybe I speak for everyone when I say it's Friday. It can wait till Monday. Enjoy your evening. Everybody have a great weekend.
Thanks so much. You too, John.
Take care.